Welcome to the Deep Dive, where we take these incredibly complex blueprints from biology and, well, we try to boil them down into something you can really grab onto.
Exactly.
Today, we're tackling a topic that it touches almost everyone's lives,
Yeah.
cancer.
Right.
But we want to move beyond just the word itself and really look at the specific playbook, the rule book for cellular rebellion.
That's the perfect way to frame it. Our mission today is to give you a detailed map of the fundamental molecular rules that normal cells have to break.
Right.
And just as important, the new capabilities they have to acquire to become a malignant tumor. We're deep diving into the hallmarks of cancer.
Okay, so before we can talk about a cell breaking the rules, we have to know what the normal rules are. What is the standard operating procedure for a healthy cell that cancer just completely ignores?
We have to establish that baseline. Think of a normal cell as let's say, a good citizen in a really tightly regulated community. There are basically four key rules that keep it in check.
Four rules. Okay, lay them out for us.
So first, normal cells have a strict, and I mean strict, dependence on growth factors. They are incredibly conservative.
Conservative how?
If they don't get that external go signal, a hormone, a cytokine, something like that, they just stop dividing. And if those signals stay away for too long, they actually initiate apoptosis. They self-destruct.
So they literally need permission to live and divide.
They absolutely do. Second rule, anchorage-dependent proliferation. This is more of a physical thing.
A physical constraint.
Yes. Cells have to be physically attached to the stuff around them, the extracellular matrix, through these proteins called integrins. It's that connection that gives them permission to divide. If they detach, they usually die.
So they have to stay rooted to their home base.
Precisely. Third, you have contact inhibition. This one's pretty intuitive. When normal cells feel that they're surrounded, that they're touching other cells on all sides,
They just stop.
they stop moving and they stop proliferating. It's a natural anti-overcrowding mechanism.
Which is why, when you get a cut on your skin, the cells grow to fill the gap, and then they just stop perfectly.
That is the exact mechanism in action.
Yep.
And then finally, the fourth rule, limited proliferative capacity. Normal cells, well, they age.
They have a finite lifespan.
A very finite lifespan. With every division, they get a little older. It comes out to about 50 to 80 doublings. We call it the Hayflick limit before they just enter senescence, permanent retirement.
That is just an amazing system of checks and balances.
It is.
But cancer, by its very definition, has to find a way around all four of those. And in 2000, researchers Hanahan and Weinberg, they synthesized all this research into 6 key capabilities, the hallmarks that a cell has to acquire. So let's start with the first two. This is the classic analogy, right? Pressing the accelerator and cutting the brake.
That's the genetic war right there. Hallmark number 1 is self-sufficiency in growth signals. And this is all driven by what we call oncogenes.
Oncogenes. It's a word we hear all the time. It sounds like something that's just inherently bad, but they start out as good genes, don't they?
They do. They begin as proto-oncogenes, completely normal genes that are involved in growth and signaling. They only become oncogenes through a gain-of-function alteration.
The gain of function.
It's like flipping a switch that gets permanently stuck in the onc position. It gives them autonomous growth signals so they no longer need that external permission slip.
What does that switch look like on a molecular level? Is it a huge change?
It can be surprisingly simple. Sometimes it's a point mutation, just one DNA letter getting changed. Or it can be bigger, like gene amplification, where the cell just makes hundreds of extra copies of that gene.
Can you give us a really concrete example of that?
The RAS gene is the classic one. It's a critical signaling protein. Normally, it waits for a signal, relays it inside the cell, and then crucially, it turns itself off very quickly.
Okay.
But in, say, human bladder cancers, a single point mutation makes RAS constitutively active. It's just always on.
Always on. What does that mean for the cell? I mean, is that energetically costly, or is it just brutally efficient rebellion?
Oh, it's brutally efficient. The cell no longer has to waste energy looking for external growth factors. It has its own internal engine that just won't shut down.
I see.
Another really powerful example is amplification of the HER2/neu receptor. This is amplified in something like 30% of human breast cancers.
30%. That's a huge number. So if you know that specific gain of function is happening, does that immediately give you a therapeutic target?
It absolutely does. Identifying HER2 amplification was a breakthrough. It allowed researchers to develop targeted therapies, monoclonal antibodies that specifically attack cells that are overexpressing that one receptor. It turns a really poor prognosis into a case for precision medicine.
Okay, perfect. So the accelerator is now welded to the floor.
Right.
Now for the brakes.
Careful.
That brings us to hallmark number 2.
Yeah.
Insensitivity to negative signals. And this is all about tumor suppressor genes.
This is the exact flip side of the coin. Unlike oncogenes, tumor suppressors are normal genes where their absence or their inhibition leads to cancer. It's a loss of function.
So these are the good guys, the superheroes getting taken out.
They are the checkpoints, the mechanics, the brakes.
Yeah.
Exactly.
And the most famous of all of these has to be P53. It's called the guardian of the genome. What makes P53 so important?
Well, P53 is like the ultimate quality control manager. When a cell senses serious stress DNA damage, low oxygen, anything like that, normal P53 activates. It slams the brakes on the cell cycle, giving the cell time to repair the damage.
And if it can't be repaired?
If the damage is too severe, P53 makes the ultimate call. It triggers apoptosis, the cell's self-destruct sequence.
So what happens when cancer takes out that manager?
The whole quality control system collapses. A cell with damaged DNA just blows right past the checkpoint and keeps dividing, passing that damage on, accumulating more and more genetic instability. And this is not a rare event. We're talking 60 to 70% of all human cancers have a P53 mutation.
Wow. Okay, and we also have another big one, the retinoblastoma gene, or ARB. How does that work as a brake?
ARB is more like a physical safety latch on the cell cycle. In its normal state, it physically binds to and holds on to a transcription factor called E2F.
And E2F is the thing that tells the cell to start dividing.
E2F is the green light. It kicks off the synthesis of all the proteins needed for DNA replication. So RB's job is to keep E2F chained up.
Okay.
When a real growth signal comes along, it causes RB to get phosphorylated tagged with phosphate groups, and that tag forces it to release E2F. E2F is now free, and the cell cycle moves forward.
So if you mutate RB,
You break the lock, E2F is always loose, and the cell is permanently ready to divide. It's what happens in the heritable form of retinoblastoma.
that makes perfect sense. So the cell has now won the battle for growth, but it still has to solve its biggest problem, which is death itself.
That's a crucial shift in strategy for the cell, moving from managing proliferation to managing its own fate. And this gets us to hallmark number 3, evasion of apoptosis.
Apoptosis programs cell death. It's the cell's, sophisticated suicide mechanism. It's clean, it's organized. It's the ultimate defense against a cell going rogue.
It is. And cancer cells have to actively fight to circumvent it. We already mentioned one way, getting rid of P53 is the biggest one. But they also learn to produce their own survival factors, like IGFs, to basically bombard themselves with don't die signals.
So if a cell manages to evade death, its next big hurdle is just time. It's running out of those doublings. Which takes us to hallmark number 4, acquiring limitless proliferative capacity.
This is the push for true biological immortality. As we said, normal cells hit that Hayflick limit because of something called a telomere.
The telomere.
It's a little protective cap of DNA at the very end of the chromosome. And every time the cell divides, that cap gets a tiny bit shorter. You can think of it like the plastic tip on a shoelace.
And when that plastic tip is gone, the whole thing frays. The cell enters senescence.
Exactly. Now, this is where the sequence of events really matters. Knocking out P53 or ARB can extend the cell's lifespan, maybe by 30 doublings, but it's not forever. To become truly immortal, the cell has to solve the physical problem of the shrinking telomeres.
So how does it do that? How does it reset its own clock?
It reactivates a very special enzyme that's normally silent in most adult cells, telomerase. Telomerase is like a specialized maintenance crew that shows up at the ends of the chromosomes. It's a reverse transcriptase, and its only job is to add those DNA repeats back onto the telomere ends, keeping them long.
So it's actively rebuilding the plastic cap on the shoelace, keeping the chromosome stable forever.
It maintains length, stability, integrity. This is so fundamental that we see it in 85 to 90% of all human tumors. Activating telomerase is, well, it's non-negotiable for a cancer cell.
That is just fascinating. So the cell is now an uncontrolled, immortal entity. But this is where it gets really interesting to me. A tumor is one thing, but it needs infrastructure. It needs a way to escape.
Right. It has solved its internal problems. Now it's facing external, logistical ones. Which brings us to hallmark number 5, sustained angiogenesis.
Angiogenesis.
Yeah.
Building its own blood supply. Why is that so critical?
Well, any tumor that wants to grow beyond, say, a couple of millimeters the size of a pinhead is going to starve. Oxygen and nutrients can't diffuse that far into a mass of cells.
So it needs to build roads.
It needs to build roads. And it does this by flipping a genetic switch and flooding the area with pro-angiogenic factors.
Such as?
The most famous one is VEGF, vascular endothelial growth factor, also FGF1 and FGF2. These chemicals are signals that tell the endothelial cells lining nearby blood vessels to start growing and sprouting new vessels directly into the tumor.
And this is a major point where medicine can fight back, right? By targeting that blood supply.
Oh, a huge therapeutic target. Drugs like bevacizumab are monoclonal antibodies designed specifically to go in, find VEGF, and neutralize it. The idea is to cut off the tumor's supply lines and starve it.
Okay, so the tumor is now large, immortal, and it has a food supply. The final, and really the most lethal step, is hallmark number 6, tissue invasion and metastasis.
This is what defines a cancer as malignant. It's the tumor escaping from where it started and traveling to form new tumors somewhere else. And to do this, it has to completely undo those rules we talked about at the very beginning.
Anchorage dependence and contact inhibition.
It has to dissolve the glue holding it in place and then literally cut a path through the surrounding tissue. This means changing its structural proteins, the integrins, and activating enzymes called proteases that just chew through the matrix.
Can you walk us through that journey, the metastatic cascade?
It is a brutal obstacle course. First, the cell has to invade and migrate through the local tissue. Then it has to get into a blood vessel. We call that intrafasation.
Right.
Then it has to survive in the bloodstream, which is a very hostile environment. Then it has to get out of the vessel somewhere else, extravasation. And only then can it even attempt to colonize the new tissue and form a secondary tumor.
And that very first step, breaking away, involves a specific mechanism, right?
Yes, a key part of it is the inactivation of a protein called E-cadherin. You can think of E-cadherin as a molecular staple holding cells together. When a cancer cell turns that gene off, it loses its connection to its neighbors and is free to migrate.
So when you put it all together,
Yeah.
it's just so clear that cancer is never one single mistake.
It's a multi-step progression. It's the accumulation of multiple genetic hits. You start with one mutation, then you move through hyperplasia, dysplasia, and finally, invasive cancer. You have to activate oncogenes, inactivate tumor suppressors, maybe break your DNA repair genes. You have to pass all 6 of these checkpoints.
So for you listening, what this all means is you now have a roadmap. You can understand why cancer is so difficult to treat and why specific therapies exist. The 6 hallmarks, they define this universal set of capabilities that give the cancer cell a massive survival advantage.
It's self-sustaining growth, evasion of death, biological immortality, and the ability to fuel and spread itself.
It's an incredible story of cellular survival, just in the worst possible context.
It is. And this framework, it also forces us to think about the sequence of events. If you look back at that really profound requirement, hallmark number 4, activating telomerase, it raises a really provocative thought.
Go on.
The cancer cell has already cut its major breaks, P53 and ARBI. It's growing like mad. Why is it still absolutely essential that it turn on this immortality enzyme? Why does it still face an aging problem?
If it's already out of control, why does the clock still matter?
Because the cell's natural aging mechanism, telomere shortening, is so fundamentally powerful that it acts as the very last barrier to full-blown unstoppable malignancy. Even after the cell has broken all the other rules, it still has to defeat its own natural lifespan. And that realization just underscores how critical telomere maintenance is as a potential therapeutic target. It's a final, essential vulnerability that cancer cannot afford to ignore.